Queen Nerdling here. Today, in this video, we're going to be discussing the endomembrane system. Let's go ahead and get started. So the first subcellular component that we're going to discuss today is called the nuclear membrane. Now, this is a porous membrane that separates the cell's DNA genome from the rest of the cellular environment. Basically, it houses the genetic makeup of the cell, or the DNA. Now, the nuclear membrane is structured to prevent the DNA from leaving. So the only genetic material that gets to leave the nucleus is going to be RNA, and specifically messenger RNA. That messenger RNA is able to leave the nucleus, and it carries with it the message from DNA. It then carries it all the way to a ribosome, which is the site of protein synthesis. Next up, the endoplasmic reticulum. So if you remember from our last lecture, we briefly discussed ribosomes. And ribosomes are found in both prokaryotic and eukaryotic cells. They're also found on the endoplasmic reticulum as well as free-floating and eukaryotic cells. So the endomembrane reticulum is a membranous channel that runs throughout the cell. It helps to produce the membrane and transport proteins and lipids. So rough endoplasmic reticulum, meaning that it has ribosomes on it, is bound with those ribosomes. The smooth endoplasmic reticulum is not covered in ribosomes, and it helps to detoxify the cell, and it also makes lipids. So the ribosome bound to the endoplasmic reticulum makes proteins that are destined for outside of the cell. Meaning that if it's synthesized on that endoplasmic reticulum at a ribosome, the protein that's synthesized is actually going to exit the cell. Many times it's going to be on that plasma membrane, and it's going to serve to help cells communicate to one another. And it does this by combining with carbohydrates in a process called glycosylation. So a carbohydrate oligosaccharide chains are found on the outer surface of the cell membrane attached to proteins. So the protein is formed at that endoplasmic reticulum, or the rough endoplasmic reticulum, and then it is glycosylated, meaning that a carbohydrate is added to the protein. And that structure, whenever that carbohydrate and the protein combine, is called a glycoprotein. Now, glycoproteins are super important when we get into cell signaling, and they function in cellular communication for hormones to be transmitted and recognized. They function in the immune system as well as reproduction, and they also help to provide structure on that plasma membrane. The carbohydrate chains are bound to the proteins in that rough endoplasmic reticulum, and then the glycoprotein is folded and transmitted in vesicles to the plasma membrane by way of the endomembrane system. Next up, this guy, the Golgi apparatus. Now, in real cells, it doesn't look as cute as this guy, but I had to show him off just to make everybody smile a little bit. So the Golgi apparatus is a series of flattened and membranous compartments that receive material from the endoplasmic reticulum, and they modify it before targeting it for delivery to other areas of the cell. Next up are lysosomes. So as far as the structure, they're basically vesicles that are filled with digestive enzymes. And in digestion, remember we are breaking components apart, or we're going to be breaking those macromolecules apart. We're digesting them. Now, if you recall from biochemistry, enzymes in those vesicles will use the process of hydrolysis or hydrolysis. So they'll use water to split larger macromolecules apart to either degrade them or to use them throughout the cell for nutrients. As far as the function, they serve a function for little stomachs of the cell. Again, they digest those macromolecules. They're also the cleanup crew. So it cleans up broken down organelles that are no longer functioning properly. Lysosomes are only found in animal cells, and they're synthesized in the rough endoplasmic reticulum. So they're also involved in a process called apoptosis. I always like to remember apoptosis is a all right? Kind of like a cell bursting. So apoptosis is programmed cell death. And this occurs in cells which are diseased, damaged, or when they've basically lived their old cell life and they're a really old cell and they just need to die. So apoptosis is programmed cell death. It involves lysosomes that release hydrolytic enzymes. And again, if you break that word down, hydro meaning water and lytic means hacha, lysing or splitting apart. So those lysosomes contain hydrolytic enzymes and acids in the cell that help to break those down and help to kill the cell. So fun fact, during embryological development in mammals, birds, we have this plate-like structure that will bud from the embryo. Digits are actually formed when apoptosis occurs between the digits. So if you look right here, these cells right here are undergoing programmed cell death, or the process of apoptosis, meaning they are going to die. Now when that happens, the tissue dies because tissues are composed of cells. So those cells die, the tissues die, and now we have actual separated digits. Now when there's a disruption in a cell signaling pathway, meaning the cells don't get the correct message and they never get the message that they need to die, then we get some different types of defects, such as this right here, where apoptosis didn't occur, and you can see that this baby did not get that separation of their digits because cell death did not occur, so it left that tissue intact between the fingers. So next up in the endomembrane system are vacuoles. Now these are found in both plants as well as animals, but they serve different functions in those two different cells. So in animal cells, like this little guy, they function like little transfer ships or food vacuoles. And they use the process of phagocytosis, phago meaning food, to fuse with lysosomes. We also use them as contractile vacuoles in freshwater protists, and they pump out excess water, and it can also serve for a function of movement in those protists as well. Now, when we get to plants, like this guy right here, as you can tell, I like playing with my stuffed cells. So in plants, vacuoles function as a central vacuole, and they store water, ions, different types of proteins, deposits from metabolic byproducts, and they can also store defensive compounds. Yes, hacha! Plants have defensive compounds as well. They also store pigments, and they help to maintain turgor pressure. Now, turgor pressure is that pressure of that vacuole pressing against the plasma membrane, which then in turn presses against the cell wall, which then in turn applies pressure, and it helps keep plants rigid and give them their structure, versus animal cells that do not have those large central vacuoles, nor do they have a cell wall. They're kind of blobbish, all right? They need different structural components to really give them their structure and build on that. So next up in the endomembrane system, and yes, the plasma or cell membrane is actually part of the endomembrane system. So this is the boundary between the cell and its environment, and it controls basically anything that goes into or out of the cell. And remember that it has these properties. It is composed of a phospholipid bilayer. So that phosphate head is going to be polar, meaning it has a charge, and it is hydrophilic or water-loving, versus the hydrophobic tails, which are composed of those lipids. The lipids are non-polar, meaning they do not have a charge, and they are hydrophobic, meaning water-fearing. So kind of putting all of this together, the endoplasmic reticulum is part of the cell's endomembrane system as well. So it's involved in the production and transport of membrane and membrane-bound proteins. So as we kind of discussed earlier, we talked about ribosomes making proteins, and we also talked about the fact that the endoplasmic reticulum, when it has ribosomes on it, is called rough ER. Now, when proteins are synthesized on ribosomes, on the rough ER, those are actually being manufactured to leave the cell and embed themselves in that plasma membrane to function for cell signaling purposes. So within the nucleus, we have DNA. DNA needs to get its message outside of the nucleus, but it can't do that by itself. So it uses messenger RNA. Now, messenger RNA transcribes the gene message from the DNA, and it brings it out to the rough endoplasmic reticulum. The ribosomes here are going to synthesize those proteins. The rough ER packages off, sends it to the Golgi apparatus, which is this guy right here. The Golgi apparatus packages and tags those proteins for destinations outside of the cell. Again, meaning that they're going to serve those purposes for cell signaling. And when I say outside the cell, they'll be on the outside of the cell, almost like these little antennas, waiting for the correct signal so it can bind with them and then have a transduction pathway and then a response eventually. So kind of a very brief recap of that process. We have DNA in the nucleus. Messenger RNA takes the message from the nucleus, out of the nucleus, to a ribosome. Now it could take that message to a free-floating ribosome, in which case that protein would remain inside the cell or internal. If that message gets taken to a ribosome that is on the endoplasmic reticulum, then that protein is going to be destined for outside of the cell. And a lot of times it'll be on the outside of the plasma membrane. It will bind with that glycoprotein. It'll become that glycoprotein because it binds with a carbohydrate. And again, it will serve as a cell signaling receptor. So we have the message, leaves as messenger RNA, goes to a ribosome, ER, Golgi packages it up, sends it out in a vesicle, bam, plasma membrane, cell signaling, awesome. So I hope that that was helpful for all of you and you learned a little bit more about the endomembrane system. For more videos like this, you can go to www.nerdlingscience.com. This is the Queen Nerdling signing off. Have a great day.